That is, it's a pleasure to open up the seventh series of seminars with Dr. David Marshall. Here's the list of the next 5 seminars, total 6 seminars, including today. We're going to end on May the 2nd. So a very good group of speakers. I hope you guys can make it to the seminars. The rest of the seminars will be all of them remotely. This is the first seminar, and this is in a hybrid format. We are here at NC State and broadcasting also to the audience remotely. So David is going to present about his work during 40 years of breeding wheat internationally. David have worked for the USDA here at NC State. He is an emeritus professor at NC State. And his breeding work was mainly centered on wheat, but he also has done breeding work in oats, barley, and triticale. He has been a key player within the Borlaug Rust initiative, for which his work and his team's work have received recognition in 2020, particularly with regards to the issue of UG99, which I think it will be great part of his presentation. David, thank you. It's a pleasure to have you here with us. Please, in the audience, we will get questions in the chat, and feel free to write your questions, okay? I'm going to start your presentation, David, in a moment. Okay. Can you help me here, Joe, please? How can I get this down window out?
Are you floating controls? So you right.
All right, thank you. Right. Here we are. I hope this works. Yes, it does.
All right. Thank you very much. Thanks for all your work.
Okay.
Good morning, everyone. What I want to do is, this is going to be some science in there, but mainly I want to tell some stories as we go along of what is going on, at least with me and some of the interactions I've had on an international scale over the past, actually, it's been about 43 years. And so what I wanted to start off with was just, to remind everyone of where the big funding is, where the big programs are for international agricultural research. And the first 5 international crop improvement research centers all came on board. IRRI was the first, International Rice Research Institute in the Philippines in 1960. And then came CIMMYT in the 66. IRRI, of course, is in the Philippines, headquarters CIMMYT in Mexico. Then came CIAT and ITA, one in Colombia and one in Nigeria, both centering on tropical agriculture. One for South America, CIAT, and the other for Africa in Nigeria. And then came ICARDA, which was not really plant improvement based, but rather plant production based, although they do have plant breeders at ICARDA. And then the year before ICARDA came on board, there was actually the finding of or formation of the CGIAR, the Consultative Group on International Agricultural Research. It was formed in 1971. They have kind of a business headquarters in Montpellier, France, but they really are a decentralized. They're more all over the map as far as that's concerned. The first 5 of these were initiated and funded by the Rockefeller Foundation and the Ford Foundation. Rockefeller Foundation probably led the way on many of these. They are the ones who actually started CIMMYT back in even before 1966. CIMMYT, of course, is the International Wheat or Maize and Wheat Improvement Center. And that actually started back in the 40s when Rockefeller funded a project called the Mexican Agricultural Project, the MAP. And he started that at the behest of some US politicians, basically, because they wanted to have more input into Mexico as far as, US influence. And the best way to do this was to do it through agriculture, which was definitely needed down there. There are currently 15 international research centers as of this year. Now, where I get interested in this is in 1982, when I got out of school finally, and my first professional job was with North American Plant Breeders. It was known at that time as NAPB, which has a different connotation today. Now it's known as the National Association of Plant Breeders. It's a completely different organization. But NAPB was a private company. We were located in Berthoud, Colorado. And we were tasked with breeding wheat and barley for the entire US and Canada. And so that's kind of a big job. And so, I went to work there, and about after the first or second week, my boss at the time, Bob Heiner, who came there from University of Minnesota, he said to me, David, I want you to get more experience in field selection, field plot technique, rust evaluations and whatnot. And so he said, I'm setting up a program for you down in Mexico. And you're going to be down there working with those guys in terms of selection at Obregon, which is in the northwestern part of Mexico. And so I said, okay. And so I went down there, and on the first day of the job, the two people who came to greet me at Obregon were Norm Borlaug and Sonia Rajaram. And Raj is, of course, the guy who we know there by that name. And they said that, Bob Heiner said that you're here for a while to learn some things. He said, let's go to the field. And so we did. And at that time, Borlaug was back in Mexico because he had been away from Mexico. But in '82, he was brought back in temporarily as a director general of CIMIC because they were kind of in between a new director. They had lost their previous director general. They were looking for a new one. And he said he would take over for a year or so. And so his place is in the field. And so we started working, and Raj, of course, is very well world-renowned. Raj has had a hand in more wheat varieties across the globe than anyone. He won the World Food Prize back in 2014, I believe. And I think they quoted there where he had released somewhere around 560 varieties a week, of which dominate the world market. So I said, okay. And so we went to work, and I did start to learn about germplasm development, germplasm sharing in particular, field plot technique, et cetera, as well as rust phenotyping. Now, at that time, the wheats that they were working on and had just released the first few were called the Veery wheats. And these wheats were the product of winter by spring crosses, which went under they were the first cycle of lines that went through what's called shuttle breeding. And what they did there was that they would make huge amounts of inoculum were put in the field there in Obregon. They would make selections in the field for rust, in particular stem rust and leaf rust. And then what would happen, they would take those selections and send them to Toluca in kind of southeast Mexico, where they had stripe rust and other diseases to select for. And then they would send them back to Obregon. And so as they advance through the generations, and you're making selection, you have all this differential disease pressure on them. And that's what they were focusing on. And that's what came out at that time that was what the Veery wheats were. And they were tremendously yield producers. Prior to Green Revolution wheats, which came out in the 50s and 60s, which before that, the yields were somewhere around 2 to 3 tons per hectare, which is in the 40 to 50 range. bushels per acre. The grain revolution wheats took that up to about 5 to 6 tons per hectare. And the Very wheats way outperformed those. They were up in the 8 to 10 tons per hectare yield. And they were very stable over environments. So at that time, working with AgriPro, I was focusing on working and I was going all over these places. Oklahoma, Kansas, Nebraska, Colorado for the hard wheats, several eastern states for the soft wheats, and then also in Manitoba and Saskatchewan. Then after that, I took a job at Texas A&M University. And there, you know, as their brochures and promotional items go, Texas is a whole other country. And that's the truth. It not only is it so large to go across, but the environments, there's at least 6 different ecological zones in Texas. And we had a really good program set up at that time, between 85 and 01, where we had a big program in Amarillo with main testing in Bushland, Vernon at Chillicothe, Dallas and the Prosper Station. And then we had close at Overton and then College Station where the university was located. And then we had testing locations, big breeding locations at McGregor, Uvalde and Beeville. And you could select for almost anything you can think of at these locations. And we did. And over that period of time, we made, I think, very good progress. We had, we had 7 varieties come out from the Bushland Station, 5 from Vernon. These are just wheats, 4 from Dallas, as well as 3 barleys and 2 oats. We had a couple come out of Overton and then a few oat varieties out of College Station. And so it was a really nice team to have working together. But all things came to an end. And so one of some of the, getting more into the specifics of some of the other international activities that I had, the first one that was funded from outside sources was a BARD grant that Lloyd Nelson and I had, where we were looking at the wild species of wheat and septoria diseases, which were important over there. And I got to go to Israel for a couple of times and then go actually on a wheat collection trip in the Negev, which is the southern part of towards Egypt in Israel. And that's where the wild wheats, the wild emmers grow in particular in that area. Unfortunately, I couldn't find any pictures of that. When I was telling these guys, when I left USDA, I had to turn over to Kent all of my data and pictures and everything else. And so I don't have any of those. And so the only ones I were able to find, you know, when you gave talks in the past at various locations and venues, there's a lot of those are actually still online. And so I was able to download some of my pictures from other talks that I had given in the past. So the second project that I had, which was a USAID funded project, was in Egypt and Sudan. And that project was very interesting, looking at primarily virus, in this case, barley yellow dwarf virus resistance in wheat. And on the left, you know, if you've been to Egypt in the Nile Valley in the Delta, a lot of the production of crops there, the postage stamp type crops, as we call them. They're just, plots of wheat and plots of, I guess, chickpea, lentils and whatnot, all interspersed. And the thing is, that makes really good environment for aphids to move back and forth. And this is a plot showing what BYDV, the virus, can do to wheat. It can very kill plants. As a matter of fact, it's spread by Rhopalocyphus paddi aphid. They have mainly the PAB strain of BYD. And then on the right here, here's our wheat plots. And you see some things that are a little different in Egypt than any other place that I've been. But we did make some progress there. There was, we established an aphid rearing lab, having both viraliferous and non-viraliferous aphids to BYD at the Agricultural Research Center in Giza. We began controlling screening of Egyptian cultivars for tolerance or resistance to BYD as well as the aphids. We established field locations in Minya, which is about in the middle of Egypt, down the Nile towards Sudan. And then we had one at Marsa Matruh, which is out in the kind of the northwestern part of Egypt near Syria. Well, not Syria, whatchamacallit? Libya. I get my country. And we made, and I made a lot of initial crosses between the wild emmers from Israel with the Fiestivum, the cultivated wheats in Egypt. It's kind of, I guess this would be a form of detente using the Israeli wild wheats to cross with the Egyptian variety. And all of those were turned over to the Egyptians for selection. So the next project that I had, and this is something that I would say to try to get all plant breeders, if you have the chance to do it, and that is to go to the center of origin of the crop. There's so much to see and learn when you go to those places. And I wanted to make sure that I was able to do that. And so this is the Fertile Crescent, as everyone knows, is this green area here, which makes up, it's kind of the borders of Israel, Egypt, Turkey, Iran, Iraq, and Syria. And that's, where a lot of farming civilization was thought to have begun. And then this other area here is where all of the diversity is typically found for wheats and barley. You find all of that in that. You find almost every relative of wheat and barley in those areas. In fact, in the present day borders of Turkey, which runs about here, on this map it's called Anatolia, I believe there are somewhere around 25 or 26 identified different species of wild wheats. And in Turkey, there are 23 of them occurring just naturally. And so it makes sense, and they're a very easy country to go to. And so it made sense to make a collection there. So this is, I was able to make two of them, one in 1991, one in 1995. And the one in 1991, I had an accomplice with me, that's Lloyd Nelson there from Overton, just to show you some of the things that you run into when you're trying to make collections in the back roads of Turkey. This is moving north of Ankara towards the Ilgaz Mountains, and we couldn't get across. It dropped down about 2 or 3 feet there. So we just had to wait a little while. Here's a field of naturally occurring Triticum monocotyl. And they just grow. The first time I did it and saw it, it was like, oh my God, these things are everywhere. And you can just go in and just find basically all the different species, almost in a single field. And then by going across the country, you get all the ones that you couldn't find. This is again a plot of this is some cultivated wheat which had intermixed with it the wild wheats, and then this is the kind of the northern Anatolian plateau, and then this is myself in I found a field of cultivated emmer, which is still a grown crop in that area. It's a diploid wheat, which is still grown. It's hulled, and so the hulls have to be removed in order to make it.
Yeah.
And so this is just an example of what we did in Turkey. This is regular wheat, Triticum domesticum, red wheat. And then we found a lot of Triticum umbellulatum. Believe it or not, these are all relatives of wheat which you can cross with normal wheat. Most of these you have to go through a embryo rescue, and we became fairly adept at doing that to get plants. But we use mainly Triticum. We made crosses with all of these: umbellulatum, cylindricum, trianciale, and monococcum. This collection became known as the TTCC for Texas Turkey Cereal Collection because we had oats and other things in there as well. I developed about 55 populations from that and distributed some of those collections as well as the populations and derived lines from that to cooperators throughout the US, Canada, and back into Turkey. And the one thing we also did in Turkey was that before we left, at each time, every head that we got seed out of, half of the seed stayed in Turkey and half of the seed came with us. That way, if they wanted anything to look at anything that we had collected, it was there. Okay. Now this one is kind of interesting because one of the things that I was interested in looking for, and then within the like cool season grasses, fescues, and things of that nature, there is an endophytic fungus which is advantageous to the plant. It renders those plants resistant to drought as well as some diseases, and it helps them grow. It's a neotyphodium fungus. And so because some of these grasses are distantly related to wheat and some of the wild species, I said, can we find this fungus in any of the wild wheats? And so I had a visiting scientist from Turkey who accompanied us on some of our trips. She was mainly an interpreter. But she came to Texas with her family and went to work looking for this fungus in the wild wheats. And so she was looking for that and she found it in two of them. Triticum tripsicoides. This is a kind of a bundled up. The head itself is like this. It looks like, like a real wild grass, but it is a Triticum species. And then Triticum dicaceans, which is more closely related, has a C genome. Going through the literature, there have been people who have crossed wheat and dicaceans with Triticum estivum. I think they went through the bridging technique of first going to a tetraploid and then going to a hexaploid in terms of crossing. But I didn't make crosses with these. But what I did in 1997 was go to New Zealand for a year. And we took a look at being able to take the fungus that Werner Tinali had found in each one of these, the neotyphodium, and to see if we could use the techniques they used in ag research in New Zealand to introduce that into Triticum estivum. And I failed completely at doing it. I made so many introductions, and because it's physically you germinate the seed, you then wait for the initial meristem, the growing point to come up, and you simply physically lay the fungus on top of it. And that's how they do it for fescues and all that. And a few times we got the plants to grow, and but then they died. And so we were unable to make that switch. So, but I thought it was a good idea. Okay, after Texas A&M, I came here, and this would be 2002 to 2021. And it was interesting. Of course, I have my own breeding program and all that, but I guess it was in maybe 2003. At that time, there were very few people in the world still working on stem rust, mainly because stem rust had been controlled with the Viri wheats and the other wheats that came after them. And what had happened is that those wheats had a translocation from a Russian winter wheat called Kavkaz. And it had the rye translocation in it. And that's what had this SR31 gene. the SR31 gene and the rye translocation just brought over a lot of nice characteristics into wheat. You had mildew resistance, you had rust resistance. It was a really nice cross. So, and that really kind of kept everything, all the stem rust populations down across the world since the late 70s until about 1998. In 1998, in Kigali, Uganda, William Liguri was going through his plots, and he saw that the lines in the rust nursery, he saw that the lines that had SR31 were hit with stem rust. And I was on there was like an email exchange that people who were involved with stem rust got, and he sent it to everyone, and he said, could everyone take a look at this? And I think everyone, including myself, responded, you must have planted wrong. Because these lines don't get stem rust. And so he said, no, I got it right. And so he sent the sample to the Rust lab two of them actually, one to Rust lab in Wageningen in the Netherlands, and one to the USDA lab in Minnesota. And I went through the testing for it, and they found that it was different. It was a completely different combination of stem rust resistance genes, or stem rust genes which could attack wheat. And then it was interesting because this combination of what it was virulent to didn't occur almost in any other type of combinations before this thing showed up. It was almost like it was almost like it just got beamed down. It was very different. It didn't have much of a lineage to anything else. And so it was just there. And so that once that information got out and it was confirmed by Zach Pretorius in South Africa, Ravi Singh, William, and Tom Payne, that it became known as PGT, or Puccinia graminis tritici, which is the fungal name for the pathogen. UG99 for Uganda 99. And that's what it is known as that from then on. The issue is that this pathogen could attack and kill basically most of the deployed stem rust resistance. And most of that resistance was based on SR31 on a worldwide basis. And so this is not something that you're going to fool around with. And it really brought people's attention. And so this is a publication that Ravi Singh put out in 2008, kind of the immediate risk areas. I don't know if you all can see those. Initially, it was about 1 billion people would have been affected. Of 117 million tons of wheat, 19% of the world wheat population could be eliminated. And this was the pie chart that he made of resistances or susceptibilities, more importantly, which is all the red that existed in those areas already. So this brought a lot of people out of the closet to take care of this, or to try to take care of it. Eventually, the disease ended up spreading all the way down to South Africa, all the countries in between here, and then started going east. So in 2005, a lot of folks got together, including myself at USDA, a few others, Eugene at the Minnesota lab. And we met with a bunch of funders as well as other scientists across the country, across the world, I should say. And we said, okay, well, we need to do something about this and screen lines kind of more systematically so we get an idea. And then CIMMYT really put together, along with the Bill and Melinda Gates Foundation, they put in, I think it was initially about 27 million to this project. And we established a testing location in Njoro, Africa, in Kenya. It was originally known as CARI, or Kenyan Agricultural Research Institute, but then I guess back in maybe 07, 08, they became known as CalRO, the Kenya Agricultural Livestock Research Organization. It's a really interesting place. The Canadians actually had a testing site there back in the 40s and 50s. And so there was some history as far as working with wheat there is concerned. It's located almost right on the equator, about 100 miles or so northwest of Nairobi. And it's in the Rift Valley, but it's at high elevation. And so it's about 8,000 feet elevation on the equator, which makes it unique in that you can grow almost every wheat in the world at that location. And that's why it was put there. The only thing we had to do with the winter wheats, and I worked as to how to better grow the winter wheats over there, was to work out a whole vernalization scheme and then a transplanting scheme, because it doesn't get cold enough to vernalize winter wheats. That's the only thing. But once you get them vernalized, they grow fine there. Each year we had, it's funny, we had this drip irrigation was initially put in, but at first there was actually hand watering because it was, that's where we started from. This is the USDA nursery, taking a look at stem rust and then the CARI and JARO sign. So I worked with CalRO and CIMMYT to optimize how to vernalize winter wheats there and transplant them to the field and get them to grow great. We went from planting rows to planting hill plots, which is much more efficient as far as getting things done there. And we worked out the protocols for winter wheats. We then organized and evaluated all of the U.S. winter wheats and barleys in JARO. I did that for each year, pretty much from 05 to 21. A couple of the years I was either sick, or we were prohibited from flying there as federal employees. The spring wheats were evaluated by a fellow that some of you know, Matt Rouse, from the Cereal Disease Lab. But in addition to doing all that evaluation, sending out the information to USDA cooperators, university and private company folks, everybody was submitting their lines for testing for this because they really wanted to know. I also crossed and selected, tested about 1,200 winter wheat germplasm lines, breeding lines, that were then distributed to breeders across the country. These lines all have multiple stem rust resistance genes in different combinations. We did do a lot of genotyping of these lines to get that done. And many of them also have leaf rust, stripe mildew, and FHP resistance. So my last project, international project, was kind of came out of the project in Africa in stem rust. Because if you remember, the map that showed where stem rust was going, it was heading right towards Pakistan and India. And there are tremendous wheat acreages in both of those countries, and a lot of it is a very subsistence type. They have a lot of production there, but if a disease like UG99 were to get into there, it would have been a tremendous issue and a lot of problems. Several of us at USDA and CIMMYT, we got to work with the Pakistan Agricultural Research Council, explained to them the problem, and then we began with funding. It was a combination of funding from USDA and USAID, where we put together the WPEP project, or the Wheat Production Enhancement Program. The objectives of it, number one, was rust surveillance. In Pakistan at the time, they didn't have a rust survey or rust surveillance of any type. And so in order to know if you have UG99, you need to be testing for it. And so that was one of the first things we set up. was a rust surveillance program. It employed a bunch of people throughout the country who were collecting samples, and then we set up a lab in a place called Marie, and I'll show you some photos of that. And then we had pre-breeding and breeding. We were heavily involved here at pre-breeding. The breeding process, then the lines we were selecting were getting sent back to Pakistan. It also involved seed production, agronomy, which was at first done by ICARTA. We brought them in mainly for political reasons, but they had a bunch of folks who were doing some work there in Pakistan. And they did a great job on some agronomic techniques. And then coordination of those scientists, particularly in Pakistan, and then capacity building for the country. And it, you know, if you compare this to other projects, it was kind of modestly funded. You know, the project ran for, it was supposed to run for 10 years. They ran for about 11 or 12, give or take. But if you take all the money together over that period of time, the money came in tranches, you know, where it was, you get this much, you show your results, then you can get the next one, and et cetera, et cetera. But it amounted to somewhere around $12 million over that period of time, which comparatively is fairly small on these international projects. But the reason why it worked was because of these locations and the people who worked here. These are the people who we brought on board, and these men and women who worked at these locations were just fantastic. Within the Pakistan Agricultural Research Council, of course, we worked with the National Coordinator Program in Islamabad, and then we worked with people at all of these locations. Kaigan up in the mountains north of Islamabad, Karachi all the way down south. Marie is where we did the greenhouse. Beta, that's in Balochistan over in the west. Gilead, again, in the mountains in the north. And then the areas, again, that we worked in the main wheat areas, these are the locations that we worked with in the Punjab. The two big ones were Faisalabad Wheat Research Institute, and then the Barani Agricultural Research Institute in Chakwal. Down in the Sindh, in the southeastern part of Pakistan, we had two locations we worked with. In the KPK, just north and east of Islamabad, we worked in Nowshera and Peshawar. And then we also had that location in Balochistan. And then we worked with a couple of folks actually up in the AGK, kind of the disputed areas up there. And they still wanted to work with us quite a bit. So like I said, the first things we did was set up a surveillance program. We had to first build them a greenhouse. And so this is the location in Marie. It's in the mountains. This is the greenhouses we added on. We had many talks over the years. I used to go kind of 2 or 3 times a year, and then looking at the final results. And you know, we set it up very similar to how the Cereal Disease Lab has their rust screening set up in Minnesota. And then there was the pre-breeding, breeding, and seed production. Of course, there was a lot of crossing going on, exchanging germplasm, et cetera, et cetera. But I used to just go over and just love to go through the plots with these guys. Right here, we're looking at some of the seed increase plots. This happens to be Mohammed Imtiaz, who was the CIMMYT in-country representative. And I had on that visit Jianming Cheng, who was a ARS stripe rust expert, because we were starting to see a lot of stripe rust in the wheat breeder there. This is at Chakwal, talking to the wheat breeder and the interpreter. And then some of the guys taking notes in some of the hill plots there in Islamabad. There's a lot on this slide, but this is, I believe, of all the projects I've worked on in the past, this is probably the most successful one. Mainly because maybe I had more experience as a PI working on this, but the cooperators were just out of this world. One of the probably take-home messages I see here is this one. Going from 2010 to 2016, 17, through crossing as well as identification of what they had already in their program. The elite Pakistani lines, this was the increase in UG-99 resistance over that period of time. We have, again, established infrastructure by the annual wheat rust, for the annual wheat rust survey. They didn't have any uniform national trials when we got over there. And this is very important to me. You need to know how your best lines are going to do everywhere in the country. And so we set up the National Uniform Trial, the NUYT as it's now called. They have an annual meeting on that, which I attended a few times. We set up the Traveling Wheat Seminar. Actually, CIMIC set up the Traveling Wheat Seminar, which is where the groups of all the breeders, pathologists, other folks who wanted to go, essentially started in Karachi and then follow the wheat crop going north and go to all the locations where these guys were to see how their lines looked at all the different locations. We did a lot of genotypic and phenotypic selection breeding lines for all the main wheat growing areas, the Punjab, Sindh, Balochistan, and KPK. We did develop a new, diverse, unique wheat germplasm group, taking the Pakistani spring wheats and the US winter wheats. And we were selecting for spring characteristics in Pakistan and winter characteristics over here. And there's still some F2, F3 lines we have in the field, or USDA has in the field here. We cooperatively identified and quickly released over 25 new rust-resistant, high-yielding wheat varieties. That's a USAID estimate. I think the number is closer to 50, between 40 and 50. And perhaps the bottom line, in this case, literally the bottom line, UG-99 stem rust was never found in Pakistan. It's impossible for us to say that we had any influence on that at all. UG-99 kind of stopped spreading. It didn't go very far north, although some of the variants were found up there because it was mutating the whole time. But it was never found in Pakistan. Thank God, I suppose. On the lower right here, we had a ceremony one year at Chuckwall, which was a big cooperator for us. And I got to plant an olive tree, which has my name on it. Okay. So for what it's worth, maybe nothing. Just, one of the things I've learned over 40 some years of working with a lot of folks around the world, you got to be optimistic. And to me, optimism means you got to pursue maybe improbable goals and still keep going. You got to be willing and determined to deal with difficulties, and you got to be confident about what you're doing. Other things, I think complacency can be dangerous in scientific research. I think trying to please everyone rarely works. Science and what we do is not a personality test here. You want to achieve and maintain a clear view of reality and try to want to have fun as well. And with that, I will stop.
Thank you, David. Please, the remote audience, write questions in the chat. And questions here from the audience, please.
Dave, do you think with this collecting trip you made to, I guess, Israel and Turkey, would that be possible these days? Because I think countries are more protective of their own sort of genetic resources.
Yeah, I mean, there was a lot of discussion even back when we went, and that was one of the reasons why we left. We didn't want to go in like robbers and just take. And so we went, we identified, and they got all the seed, the same seed that we had, as well as the information. Yeah, Christine.
Very, very important Pakistan project there. Great work. What do you think accounts for the sort of stalling out of UG-99? I mean, do we have an idea about where it is now? Were these strains with these strange new mutations not as fit as the non-mutated strains?
Yeah, some were not fit at all. In fact, back in, oh geez, it was probably '06, I think, Eugene identified a strain of UG99 that attacked SR36, which is one of our major genes that we were working with. And he found it once in the nursery. And then we blew it up and we then introduced it in the mixture of races that were being inoculated at Injaro. And it never survived.
Not competitive.
Some of the other mutants, like the SR24 mutant, was extremely well adapted. And we went back and some of the lines that we identified as being completely resistant in the first 2 years, in year 3 were completely susceptible. Now, why it kind of stopped spread, no one knows the exact reasons why. It still exists. The original UG99, my understanding is that in surveys in Kenya and Ethiopia and even down into South Africa, they can't find the original. They can find some of the mutants that came in. But it's still, during that period of time, all of those countries have released resistant varieties. So,
And so maybe the strains just aren't as competitive on resistant varieties or on other varieties as the non-UG99.
I mean, there was a lot of effort putting in, not only by us, but for other people to get resistances out as quickly as possible. And so hopefully that's some sort of an effect, but we'll never know.
David, as a homage to your work, there's greetings from Pakistan here in Kenya.
Right.
So at the peak of the seminar, we had 105 remote attendants. There's a couple of questions, if you allow me, Gina, sorry. One is, beyond Kenya, how widespread is UG99 currently? And you just talk about that, which countries are now more prevalent? Is it more prevalent? That's one question. There's another one later. Go ahead.
No,
You already talked about that.
Yeah.
Yeah. Why UG 99 was special, so much funds allocated, although it's just a new race like any other race that happens very often, stripe and leaf rust.
yeah, well, mainly because of its potential to just kill plants outright and to do it very rapidly. The stem rust pathogen produces much more progeny than either one of those other ones. And so for every single infection, you get more spores produced by the stem rust pathogen than you do for either one of the other ones. And so it has the greatest potential to cause epidemics. And of course, it's spread by the wind. So you're not going to put up a wall and keep it out. And so it has great potential to cause epidemics if it gets going.
Gina?
Yeah, I was just gonna ask, do you actually get surveillance data like from Russia and China and other countries further? I mean, is there good surveillance data available from all of the countries at this point, or?
It's spotty, and even from year to year, it's spotty. Some years you'll get some good data, and other years you'll ask for it, and that will be the last you heard about it. So it's the point is that there has been other countries, especially the kind of southern or like the countries along the northern Mediterranean, who do very good surveys, and they have never found it. Okay.
I'm just wondering, to what extent the limit of the spread is related to climate? And if climate's is shifting around, whether it's possible that this could appear in new places.
Yeah, well, undoubtedly it could. It's interesting that of the 3 main rusts on wheat, stem rust is the one more adapted to higher temperatures than any of the others. Leaf rust is kind of a middle temperature type rust. Striped rust was up until 2000, a kind of a more cool temperature rust. It occurred like in the US, mainly in the Pacific Northwest. But as of 2000, 2001, we started to see striped rust throughout the US. We always saw a little bit of it, but I mean, it came in big time. There were new races of striped rust at that time as well. I think it may be pushing it to say this is due to climate change, but, you know, but certainly, I mean, that's the fact.
Yeah, I was thinking not so much that it's pushing it, but the fact is the climate is changing, whether that's gonna open up some opportunities.
And yeah, I agree with that. And also the fact that some countries, like in more tropical countries, are trying to be more self-sufficient in wheat, and they're
Yeah.
promoting. I mean, Nigeria, from time to time, they get this big initiative to be self-sufficient in wheat, and it fails, and then it comes back again. What is the current status of the nursery in Kenya? Is it continuous? That's a question here.
Yes, there is a USDA scientist in Nebraska who is heading that up now, and for the life of me, I can't think of his name.
Jeff Bone, right?
But he has taken over the nursery when I stopped.
And one of the questions here is Ethiopia being in the path of UG 99. Why is that? It was not a major issue there. Is that most of the varieties were resistant?
No, it was an issue.
It was an issue.
Yeah. And then, in fact, you know, they are, Matt Rouse, again, who did the spring wheat evaluation, spent more of his time in Ethiopia than I did in Kenya.
Any other question? We do have a couple of sort of recognitions for David. Please stay if you're remotely participating. Any other questions here from the audience? If not, I want to invite. Thank you so much for everything, David, that you have done. It's greatly appreciated. Okay. Simon, you want to come up? There's a recognition from our graduate students, and then a recognition from the USDA itself.
Great.
You can speak to the.
Yeah, I'll speak to the mic. Yeah, and I couldn't think of a better day to do this. So we started a new award this year, the Plant Breeding Club. This is a graduate student plant breeding club, although we have some undergraduates involved as well. And we wanted to start a career recognition award that specifically came from the graduate students of this organization. So with that, I'd like to present the NCSU Plant Breeding Consortium Graduate Student Career Recognition Award. And it reads, recognizing a lifetime of mentorship for Plant Breeding Consortium students and a prosperous career of scientific innovation. Thank you for your time.
All right.
This is getting to be a bit much.
Yeah.
Well, for the audience online, my name is Mukarrab Qureshi, and I'm the USDA ARS location coordinator here in Raleigh, North Carolina. David and I have gone way, way, way back. I remember the days when we were talking about it, taking our Federal Executive Institute training together on a day which was the most snowy day in Charlottesville, North Carolina, Virginia. But David, on behalf of my colleagues who are here, we wanted to thank you for your years of service to USDA, to ARS, and to our nation. And for that recognition, USDA is offering you a certificate of appreciation. It says awarded to Dr. David Marshall for your outstanding work spanning several decades in the US and across the globe. Your groundbreaking contributions in wheat breeding around the world have earned you the title of the pioneer of a second green revolution. You have made all your Raleigh, North Carolina Agricultural Research Service colleagues and the location so proud. Thank you for your service. What you did was important, and you made a difference. And
Or
Most of you know that USDA is in the process of building a new building at NC State campus. It's called Plant Improvement Facility. It's a congressionally appropriated funding which is making this possible. And when this building was conceived, David was the brain behind it. It was his vision that we needed this type of facility here to do federally funded agricultural research. Of course, as luck would have it, things got delayed and delayed and delayed. And David, after 5 years, we still haven't. The building is there, but it's still on papers. But if it was in my power, I would name that building as Marshall Building. But as federal government rules say, we cannot name building after employees. But David, I'm going to present you the shovel, which we use actually to break the ground for this new building. It has USDA and NC State monograms on it. And it has a visual of what you had originally planned, the design and the picture of this. So this was the shovel. I cleaned it up for you. See the dirt, but I want you to keep this as our personal thanks to you for your leadership to have this research facility for generations to come.
Thank you so much.
Thank you so much, David. Thank you, Farah.
Yeah.
Okay. For those of you in line here, we will meet again next Thursday to talk about banana breeding. Presentation from Ronnie Sweeney coming from Uganda in IITA in Uganda. Okay. Thank you so much for your participation. Thank you, Peter and Farah, for helping put this together. Thank you, David, so much. I look forward to staying in contact with you. All right. Thank you.